Hello, everyone. Welcome to today's live broadcast entitled Miniaturized Spectroscopy for Biomedical Applications. I'm Jeannie Lakey-Northrup, Senior Custom Content Manager of Spectroscopy. I'll be your moderator for today's event. We are pleased to bring you today's webcast presented by Spectroscopy and sponsored by Hamamatsu Photonics. Before we begin with today's broadcast, I just have a statement to share from our sponsor. Hamamatsu Photonics is a leading global manufacturer of optoelectronic components and systems. Some of these include sensors and systems for spectroscopy, including ultrafast, scientific-grade cameras, beam monitoring systems, photon counting detectors and systems, photomultipliers, photodiodes, and IR detectors. Before we begin with today's broadcast, I also want to remind our audiences that this webcast is designed to be interactive, and so we encourage you to ask any questions you have throughout the event. We have an awesome presenter on today, and I look forward to joining him during the Q&A session later. For any questions you have, you can submit them through the Q&A box. That box is located at the bottom of your video player. For viewing purposes, you can enlarge your slide window by clicking on the small icon located in the bottom right corner of the media player. Please note, however, that all slides will advance automatically throughout the event. If you do happen to experience any technical problems viewing or hearing today's presentation, please click on the question mark help widget that you see located in the top right of your presentation window. Now, I'd like to introduce and bring in Gaetano Panagia. Gaetano serves as a technical marketing engineer at Hamamatsu Photonics. He's responsible for curating technical content and market research on mid-IR detectors and spectrometers, specializing in GaN sensing, agrophotonics, and food sorting applications. He contributes to Hamamatsu's pilot line service, aiding customers with their custom optoelectronic modules. Gaetano, thank you so much for your time today. Appreciate you joining us and providing such an international perspective.
Thank you, Jeannie, for the kind introduction. I'm very happy to be here, so welcome everyone in this presentation about miniaturized spectroscopy and its medical applications. I am Gaetano Panagia, Technical Marketing Engineer with Hamamatsu Photonics Europe. In my next slide, I want to show you the outline of today's presentation. First, I want to give you a brief introduction about Hamamatsu Photonics, and then we'll talk about spectroscopy techniques and their medical applications. So I will give you a list of several spectroscopy techniques, and I'll show you for each of them an application in the medical field. We'll do the same thing for light sources briefly. And finally, I want to explain to you, I want to show you what the Hamamatsu pilot line service is about and how with this service we can help bridge the gap between medical research, medical applications, and photonics technology. So let's start first with an introduction about Hamamatsu Photonics. So Hamamatsu is a company who has a long history in the optoelectronics manufacturing market. We are there since 1953. The company is headquartered in Japan. We have about 6,000 employees. But what is most interesting from this slide is the number of products that we currently have in the market, which is 15,000 products. And most of this is custom specific. So you should not hesitate coming to us with your custom requirements because we are capable to make it happen. What Hamamatsu produces, optoelectronic components, meaning light detectors, light sources, some of which we'll see today, but also complete systems like camera systems or systems used for digital pathology, for semiconductor testing. Today, we'll talk mostly solely about medical applications of Hamamatsu products, but I won't be able to cover everything. So I will talk specifically about spectroscopy, but I just want to, I want you to make you aware about some interesting products beyond spectroscopy briefly. So we are a market leader in PET applications and digital radiography. For PET, we offer photomultipliers and MPPCs, so multi-pixel photon counters, which are detectors sensitive to very faint lights, which are coupled to scintillators to detect gamma rays that are used in this kind of diagnostic instruments. On the other side, we are experts in digital radiography, where we supply the market with scintillators again, but this time for X-ray sources, for X-rays. So we also provide X-ray sources and cameras of many kinds. But today's presentation, as I said, is going to be about spectroscopy. So let me start with one of the most basic explanation, one of the most simple explanation of absorption spectroscopy, which is the dispersive spectroscopy. How does that work? We have a light source, which is generally a broadband light source. The light is sent through a sample, and then when the light is transmitted from this sample, it gets some absorption spectrum. So basically, the wavelengths that are absorbed by the sample are very characteristic for the sample, for its composition, and also the concentration of its components. So analyzing the light that comes out of the sample using a spectrometer, we are able to observe each wavelength component of the light, and we are able to characterize the sample itself. So the light that comes out of the sample is passed through a transmission grating where it's split into its wavelength components. Each wavelength component is then focused on a different pixel of an image sensor. So this is a general structure of a spectrometer. You're seeing here a transmission measurement. We can also measure the concentration of some substances in the sample using Lambert-Beer's law that you see at the bottom of this slide. So we measure the absorbance, and then knowing the molar absorptivity of the substance that we are looking for, and also knowing the optical path length, so the length that the light has to cross through the sample, we are then able to measure the concentration of the specific compound we are looking for. But what is a possible application of this technique in the medical field? Well, that can be applied to portable and non-invasive blood analysis. And for this, we're using visible to near infrared spectroscopy, so wavelengths that go between 350 and 850 nanometers. Simply putting a finger through this analyzer, you can get in 50 seconds an estimation of the amount of hemoglobin, bilirubin, oxygen saturation, creatinine, blood glucose with up to 93% accuracy. So this information is taken from a patent, so from a real world example, from a real world product, which uses white LEDs for illumination and allows us to make low-cost instruments that can be portable, battery-powered, and it's perfect to be used in remote areas with limited access to medical facilities or trained personnel to do screenings. For example, it can be used for rapid screening of anemia. This is also a real-time measurement, so while usually with blood samples, you need to take the sample and send it to a lab. In this case, the measurement is done in 60 seconds and in real time. And this is possible thanks to Hamamatsu spectrometers. In fact, to the left side of this slide, you can see the most compact visible spectrometer in the market, which is about the size of a fingertip. We have several versions available. In the near visible is the one that enable this blood measurement, and then we have another version in the UV sensitive from 190 to 400 nanometers. The resolution of this system depends on the wavelength range. In fact, at the UV, you can get 8 nanometers, while in the near visible, you get 15 nanometers. And you can see also the scheme, the structure of this device, whose fabrication was possible only thanks to our special CMOS process, which allowed us to make an input slit of the spectrometer in the same structure of the CMOS image sensor. So we were able to make the device as compact as it is. This device is an analog output device, so it needs some PCB, some interface. It needs to be integrated with electronics. But then on the right side, you can see the integrated version that we also offer, that we call modules. These are devices that can be sensitive in a wide range of wavelengths between 200 and 2250 nanometers with different ranges. So each product has some range in this bigger range. We have several resolutions available, and this is an integrated solution with a USB interface. And we're also working on offering an Ethernet interface. It can be cooled whenever you need higher sensitivity, or can be uncooled whenever you need a compact solution with low power consumption. Absorption spectroscopy, though, it's not only done using diffractive spectrometers. There is other techniques, and there is one which is called Fourier transform spectroscopy. So I show here a schematic of it. In this case, we have a reflection measurement. So we're not measuring the light transmitted from the sample, but we're measuring the light that the sample reflects. And this is typical with solid samples or powders, while the other technique is more common with liquid samples. So light reflected from the sample gets into this device where it finds a beam splitter. So here the red beam is split in two different arms. One, the green one, bounces off a fixed mirror, while the other one, the blue one, is directed towards a MEMS mirror, which is a movable mirror that moves up and down. Whenever these two light paths recombine, they generate what we call an interferogram, which is shown at the bottom left of this slide. And this is created at the InGaAs photodiode. So this is the shortwave infrared signal that the photodiode records. When we run a fast Fourier transform algorithm on this interferogram, that's when we get the absorption spectrum of the sample, so the spectral information that we were looking for. And here you can see the spectral information from several substances like caffeine, acetaminophen, aspirin, and theophylline. And you can see how these have a characteristic spectra and with different intensity and different positions. So basically, we are able to understand and characterize the sample using this technique. And among all samples that we could use, we have the possibility to run it through urine. And in that case, we can get very interesting information like we see in this slide. In the graph, you can see the Fourier transform spectrum of some urine got from healthy patients. And then this was compared with the urine spectra obtained from patients with pathologies like hematuria and pyuria. You can see how there is a large difference both in the intensity of the peaks and the position of the peaks between the control sediment and the pathological spectra. So this technique can be used not only to identify specific substances, but more simply, it can just be used to identify shifts from a normal or from a typical spectrum in order to identify very early some signs of malfunction in our organism. For example, this technique has been used to classify endometrial and ovarian cancer, specifically observing the peaks generated by proteins and nucleic acids. Hamamatsu has a product for this kind of technique that you see in this slide. So you can see to the left the schematic that I showed before, but in this case, we also show some additional components that are integrated into this module. For example, we have a laser, which is called a VCSEL, and a silicon photodiode. These are used to track the exact position of the movable mirror in order to obtain a precise measurement of the absorption spectrum. And you can also have a sneak peek on the inside of this MEMS mirror, so this movable mirror, which is fabricated with our special process, and that moves up and down to generate the interferogram. The device itself, it fits on the palm of one hand. It is sensitive in the shortwave infrared, so from 1100 to 2500 nanometers. It is pretty quick in its analysis, 175 frames per second. But most importantly, when we compare the Fourier transform spectrometer with your previous diffractive spectrometer, this device does not compromise between resolution and signal-to-noise ratio. In fact, diffractive spectrometry is characterized by a trade-off between the resolution you can achieve and the amount of light that gets into the sensor. So in turn, this becomes the signal-to-noise ratio that you can achieve from your measurement. While this kind of analysis, so Fourier transform spectrometers, they do not go under this compromise. This device, again, is an integrated module, and it is offered with an Ethernet interface. Moving from absorption spectroscopy to a completely different technique now, which is getting a lot of popularity nowadays, and it's called Raman spectroscopy, and it works in a completely different way. While before our light source was broadband, in this case, we're using a laser, which is characterized by a very narrow single wavelength emission and a pretty powerful one. So we direct this monochromatic light towards our sample, and there is two kinds of interactions that can happen. The most common, it's called Rayleigh scattering. So the light, it's sometimes deviated, so scattered, but it keeps the same wavelength as the laser source. But 1 on 10 million photons, so a very low number of the photons that the laser emits, shifts wavelengths. So it changes wavelengths going up or going down. And we can see this in the Raman spectrum of carbon tetrachloride that I included in this slide. You can see that the Rayleigh scattering signal is very high. It lies above this graph. And then there is very small peaks shifted to the right or to the left compared to the original emission wavelength of the laser. And these peaks, they give us information about present chemical bonds in the sample, crystallization state of the material, so overall material composition and component concentration. And here you also have a schematic of a possible Raman module in which you have LED module, so LED laser diode module, which sends the monochromatic light through the sample passing through a beam splitter. Light reflected from the sample gets into the spectrometer, and it's analyzed, again, wavelength by wavelength, observing this Raman scattering phenomenon. It is very important to notice that to build a Raman spectrometer, you need a sensor which is extremely sensitive, because the signal, the Raman signal, as we said, is very faint compared to the Rayleigh scattering signal. The application of Raman spectroscopy that I want to show you are again in urine analysis. In fact, we can characterize again the spectrum of the general looking spectrum of urine using Raman spectroscopy. We will obtain a graph like the one on the top. And this graph, this information can be used, and it was used in literature, to determine the probability of rejection during a kidney transplant compared with a real biopsy with 95% of accuracy. So the researchers were able to understand without doing a biopsy, which is pretty invasive, if a kidney transplant could be accepted or rejected by its donor. This technique was also used to detect drugs of abuse, so in forensic science, for example, detect therapeutic agents or doping substances in sports medicine, for example. The same technique can be used, a little variation of it, which needs some sample preparation, which is called surface enhanced Raman spectroscopy, was also used um, to determine if a patient was suspected or not from coronary heart disease, still completely non-invasively through their urine samples. And again, for this technique, Hamamatsu as a solution, it is a very compact module that again is miniaturized in the sense that it fits in the palm of your hand. It is sensitive in the range of 200 to 2,500 centimeters to the -1, and it has an integrated laser. So it comes with a 784 nanometers laser that emits a maximum power of 200 milliwatts. The last spectroscopy detection technique that I want to show moves from the spectrum in the visible, from the visible to near infrared spectrum, from the shortwave infrared spectrum to the mid-infrared spectrum, which means that we are now working between 3 to 10 micrometers of wavelength. And these wavelengths are extremely useful in the gas analysis, so in gas sensing and gas analysis. Specifically, we use two techniques to detect gases like CO2, that is shown in this slide. One of them is called non-dispersive infrared spectroscopy. It is extremely simple, low cost, and it can be designed with very low power consumption for battery-powered devices. It consists of a light emitter, which is generally a LED or a broadband lamp. Then you have a gas chamber in which the sample of the gas is introduced. And on the other side, you have a mid-infrared detector with a bandpass filter. So the passing band of the filter in front of the detector is the same absorption band of the gas. So whenever there is no gas in the chamber, the signal emitted from the light source and detected from the detector is pretty high. Then once the gas is introduced into the chamber, this signal decreases because some of the signal has been absorbed by the specific gas. Very similarly, we have laser absorption spectroscopy. The only difference here is that instead of the LED or lamp as emitter, we're using a laser. So again, narrow band emission with high power. And this gives to this measurement technique high sensitivity and high specificity, which means that we are able to target very narrowly a specific gas, which in this case is again CO2. But what are the applications? So why are we interested in CO2 in the medical field? The main application of CO2 sensing in the medical field is called capnography. So it's the measurement of CO2 from exhaled breath. This gives us information about cardiac output and pulmonary blood flow. So the signal that is produced, so if we observe the emission of CO2 from exhaled breath, we get a graph similar to the one that you see here in this slide. And this graph gives us interesting indications, specifically at the highest peak here, it's called end-tidal CO2 signal. And this can be related to the alveolar concentration of CO2, which is an indication of how good the exchange between our blood flow and our lungs, the air in our lungs, is working. This technique is specifically used in reanimation or in the surgical room to check if the endotracheal tubes, which are used for artificial respiration, are correctly placed inside the patient. But it can also give indication about pathologies. For example, if the capnograph does not look exactly like in this slide, it can be a signal for mechanical airway obstruction, bronchospasm, emphysema, or hypothermia. And these devices can be extremely compact. You can see it in the slide as well. This is a commercial device. It's based on non-dispersive infrared that I presented in the previous slide, and you can see how small it can get. For these applications, Hamamatsu offers mid-infrared detectors, which are made of a special material called indium arsenide antimonide, or INASP. This material is sensitive in the range that goes from 3 to 11 micrometers, which works well for CO2 because CO2 absorbs at 4.26, 4.3 micrometers. These devices work at room temperature. This means that they do not need any cooling, so we can have low power consumption while driving them. They have a linear response, which means that we can detect low quantities of CO2, but also very high quantities of CO2 without sensor saturation. They are pretty fast, so they have a rise time around 3 nanoseconds. So they are able to track quite accurately over time the changes in exhaled CO2. They are available in several packages according to the specific application. And one of the most important things, they do not contain any hazardous material. So traditionally, mid-infrared sensing is done using substances like in the detectors like lead, mercury, or cadmium. These substances are harmful to the environment when they are discarded. So that is the reason why we decided to develop a different material, indium arsenide antimonide, which does not contain any of these harmful substances. So this was the overview about the sensing technology that can be used for medical application, but you've noticed that each of the applications that we mentioned also included a light source. So briefly, I want to show you what light sources are available in the market. In several cases, when we talked about Fourier transform, when we talked about dispersive spectroscopy, we mentioned broadband light sources. These are generally made using noble gases like xenon flash lamps, deuterium lamps, and mercury lamps. They are characterized by a broadband emission from the UV to the near infrared. They are overall stable, and they can reach high intensity or high peak intensity in case they are flashed. They are generally used for in vitro diagnostics, so in the machinery in which the clinicians put the vials, for example, blood vials or urine vials. So they are generally used, widely used with dispersive spectroscopy. LEDs, on the other side, are compact sources. They're characterized by low power consumption. They have emission ranges from the visible, from the UV to the visible to the mid-infrared. So Hamamatsu offers red, infrared, and mid-infrared LEDs. They are used in non-dispersive infrared for gas sensing, but they're also, as we've seen, integrated into non-invasive blood analysis devices, like this pulse oximeter that you can see in the slide. And finally, we do offer laser sources characterized by compactness, high power of emission, and even narrower emission wavelength compared to the LEDs. As we've seen, these are necessary when we do laser absorption spectroscopy for CO2 sensing, but also they are needed for Raman spectroscopy applications. And showing a pretty interesting application of light sources in non-invasive glucose analysis. This is a topic that has been trending in the last probably 10 years. There is a lot of companies out there trying to measure glucose non-invasively and continuously, specifically for customers who are affected by diabetes. And the researchers that published this paper, which are actually researchers from Hamamatsu Photonics, tried to measure glucose non-invasively using a QCL. So QCL stands for quantum cascade laser. So this is a narrow band light source that emits in the mid-infrared. Between 3 to 11 micrometers again, and it emits at this is a pulsed solution. This is a pulsed laser. So the emission looks a little bit like a comb, like you can see here in the middle of the slide. And if you couple this laser source with a MEMS grating, which is a device that's rotating, so when the MEMS grating is hit by the laser at a specific wavelength, it is able to shift its wavelength according to the specific angle at which the laser is hitting it. So modulating the phase between the emission of the QCL and the rotation of the MEMS grating, we were able to obtain a continuous spectrum that can be used for spectroscopy, but it can have the high power of the laser and the emission wavelength in the mid-infrared. So this light source was coupled with a spectrometer that uses our mid-infrared detectors. And first, it was used to observe the absorption spectrum of glucose in a solution of water, just to see which were the absorption peaks of glucose in water. And this was also compared with a Fourier transform measurement, just to see how these two measurements compared. And after that, it was used to measure glucose non-invasively. That was done coupling the light source with an optical fiber. The light is driven by the optical fiber into ATR prism. ATR stands for attenuated total reflection prism. This is a device that is basically used to increase the interaction between light and its surrounding environment. And then the patient would put its lips around the surface of the ATR prism, so the light could interact with the lips, and then the ATR is again coupled through a fiber into a spectrometer. So we could analyze the spectrum of the light that went into contact with the lips of the patient, and we could correlate the absorption spectrum of glucose with what was observed through the lips. So in principle, it's possible to measure non-invasively glucose just putting this ATR prism and this device in between the lips of a patient. Concluding, it is quite clear at this point that each application in the medical field, but not only in the medical field, that involves photonics requires a light emission component and a light detection component, and generally these need to be put together in an optoelectronic module. Very often, this optoelectronic module needs to be designed specifically to the application. And that's what we want to do with Hamamatsu pilot line. So we know that often the people who are doing clinical research, medical research, are extremely focused on their research or on their application, but they may not be knowledgeable or they may not have the capability to put together light emission and light detection in a module in the most efficient way. And that's why Hamamatsu pilot line is born. So it's pretty clear by this point that we at Hamamatsu can offer several different light emission components and light detectors. And with the Hamamatsu pilot line, we want to hear about your application to develop application-specific prototypes. we want to develop these prototypes together with you and optimize their design, always having a final goal in mind, which is mass manufacturing. So if you are working on a specific project in the medical field and you would like to know what are the possible solutions that photonics can offer, a very good idea would be to contact us at pilotline@hamamatsu.eu, so we can start and discuss, and we can start thinking about what solution photonics can offer to your application. So with that, thank you so much for listening in, and I think we may have time for some questions, right, Jean?
We sure do. Thank you. Such an awesome presentation. It's so crazy to see how far the field has come in terms of measuring everything you were highlighting today. I mean, I'm wearing my iWatch right now, and you were talking, and I just kept looking down at my watch, thinking, wow, the value and the significance of wearables in helping manage patients and improve healthcare for everyone. Fantastic presentation. Thank you so much. With that, we are going to get ready to hop into our Q&A session. For anyone who has not done so but still has some questions that you'd like to ask Gaetano, don't hesitate to continue to submit any questions you have through the Q&A box. Again, that's directly below your video player. In the interest of time, we may not have an opportunity to respond to your question live. Have no fear, Gaetano will also be reviewing and responding to your questions after today's presentation has concluded. Gaetano, what makes Hamamatsu's components particularly suitable for medical applications? I know you highlighted it at the end. Anyone interested in partnering should reach out, but what exactly should we know here?
Right. So medical applications are very specific, very peculiar kind of applications, mostly because the development cycle of a medical product is pretty long. So they start from basic research, from medical research, then you go into clinical trials, and maybe 5 or 6 years may pass before you obtain the permissions to get out with a product, right? So in looking for a partner for a medical product, I think it is important that, of course, the partner may offer reliable products, the highest possible fabrication standards, but it's also important that the partner, like Hamamatsu does, is going to be there for with that product for 10, 15 years. So I think it's crucial for these applications to partner up with somebody who can make sure that their product, their component in this case, can be available for the whole development cycle and market cycle of a medical product. I think that could be one very important aspect to take into account.
Absolutely, very crucial. So you highlighted a bunch of different spec techniques today. You also alluded to some really great examples of their clinical significance. Which technique specifically, if you had to pick one, do you feel has the greatest near-term clinical impact as you start to project the future?
Right. I think among the techniques that I showed today, there is at least two which are already fairly quite used in the medical field, and these are the capnography one, which is widely used again in the surgical rooms or in the during reanimation of patients. So that this is already a reality. And then you have the pulse oximeters. So the one that we showed, that basically the devices that are used to measure the amount of oxygenated and deoxygenated hemoglobin. So they measure the capability of the blood to keep oxygen. So these two techniques are already widely used in the market, so they are already a reality. For the others, we are looking at introduction. For example, ramen that we've discussed, they are still in the research field, but I hope that they will become products soon.
That said, my next question was just about what's holding them back here. What do we really need in terms of addressing some of the current limitations to overcome some of the challenges for non-invasive spec methods?
Yes. The main challenges that I see to introduce more and more non-invasive methods in the field, so in our everyday lives, one is probably the size of the devices, which need to get smaller and to consume less power in order to be available in more locations. So not only in the large hospitals, but also maybe at home, in your watch, as you said. And then it's the price, the cost of it. So most of this technology faces a chicken and egg problem. So you have, until it is widely adopted, prices are still high, but then once the technology becomes widespread, prices go down and the technology becomes more democratic, let's say, then it spreads and everybody then can afford to get their non-invasive diagnosis from a wristwatch.
So you mentioned size and you mentioned efficacy. How does Hamamatsu approach this balance between miniaturization and performance for medical equipment and medical spectroscopy with the techniques?
This is a good question. So this is not only true, of course, for medical applications. It would be true for any kind of applications. But the important thing is here that we do not need to compromise between size and like sensitivity of these devices. So having control, so being from the fact that Hamamatsu has control through from every component of a spectrometer, being for the light source, the diffractive grating, the image sensor. So having control about all and the fabrication of all of them. With our knowledge in optics and photonics, we were able to miniaturize this solution in the most effective way. And that's why it was possible to create such small devices. So in the end, we want to find less and less compromises, so as few compromises as possible when designing these devices and making them small.
Gaetano, thank you so much for your time and your presentation today and all of your insight. We are going to get ready to wrap up. I have one final question that I thought I'd hold off on till the end. I thought it was a great way to wrap up the discussion. All right, taking out your crystal ball here. What emerging applications in medical diagnostics do you feel you're most excited about? You've had a really great career in this field. I'm thinking more near term, next 5 to 10 years.
Yes, it's an interesting question. I think personally, that's what I'm most excited about is this urine sensing that we've been discussed. We've talked about a few techniques. The devices are still palm-sized, but if we were able to get them much smaller, so a fractional size of that, we could be able, but personally, I take my blood test maybe once every 2 years. So having the possibility of having the test, so blood test or urine tested maybe every day, multiple times a day, I think it could be a great improvement for our society because we could be able to detect, to diagnose illnesses like cancer very early in the process, and that would help a lot with fighting against these illnesses. So what I'm most excited about would be that this kind of technologies get as widespread as possible and that everyone can monitor their health multiple times per day. It can be through urine, for example, so that we are able to fight against illnesses as early as possible.
So screening and preventive measures, really cool. So thank you so much. Gaetano, I want to thank you again for your time today. I also want to thank our audience. We had a really great crowd join us today. We've had additional questions populating in. Again, Gaetano will review and respond to your questions after this broadcast has concluded. But I want to thank you so much for your participation and for joining us today as well. And of course, I'd like to thank Hamamatsu Photonics for making today's webcast possible. We are going to get ready to conclude at this time, but as we do, I'd like to ask everyone in the audience to participate in a brief survey. The survey is going to appear on your screen after the presentation has concluded. Greatly appreciate your insight. You will also receive an email alerting you when today's webcast is available for replay, and we ask you to help forward that announcement along to anyone who may have missed today's live event but may find this interesting as well. So again, thank you to everyone for joining, and we hope to see you again soon. Have a great rest of your day.
Thank you.